Hello, and welcome to the Clinical Care Options Infectious Disease Podcast. I'm your host, Tiffany Hensley McBain. Today's episode is part 13 of an ongoing series entitled COVID-19 Answering the Questions. This series features brief updates on the latest incidents and clinical data related to COVID-19 diagnosis, prevention, and management, each followed by an in-depth question and answer session designed to address infectious disease specialists' most pressing COVID-19 questions. During this episode, Professor Jens Lundgren from Copenhagen, Denmark, will provide an update on monoclonal antibody treatments for COVID-19 and discuss the recently released information on the efficacy of leading COVID-19 vaccine candidates. For more information about Professor Lundgren and for a link to additional online education from CCO's COVID-19 Comprehensive Resource Center, please visit the show notes for this episode. Now let's get started and hear a brief COVID-19 update and answers to clinician questions by Professor Lundgren.
Thank you very much. And hi, everybody. I hope you're well, although I'm sure we all are quite busy at the moment. As you know, we have a pandemic, and I've had that since March of this year, and it's coming up to a fairly astounding number of people who's been infected with this virus that probably jumped from bats to humans around a year ago. And unfortunate also a increasing number of deaths. So we are in dire need of better treatments and also a vaccine, and I'm sure we can all agree on that. Let me just review some recent treatment updates. In particular, the first publication of what we call a neutralizing monoclonal antibody developed in collaboration between NIH and Eli Lilly. This antibody is binding to an epitope on the spike protein and serves to prevent binding between the receptor binding domain of the spike protein and the ACE2 receptor on the cell surface. Recently, in New England Journal, the first preliminary results was published from a randomized double-blind placebo-controlled phase 2 trial assessing this monoclonal antibody in patients with mild to moderate COVID-19. Of note, these were patients that were not in need for hospitalization. The primary endpoint in this phase 2 trial was biokinetic changes at day 11 after enrollment. And if you look at the table below, you can see that 3 doses of the monoclonal antibody was used, 7,000 to 2,800 and 700 milligram, and then a placebo group. And if you look at across all the 4 groups, there was a reduction in viral load between entry and day 11, as you would expect, also in the placebo arm. But there was a trend for further, more rapid decline for those patients who were dosed with a monoclonal antibody compared to placebo. Not very profound, of course, log difference of 0.22 overall, which was significant. This was a phase 2 trial, and this was the primary endpoint, but the investigators were also able to investigate how many of these outpatients were in need of hospitalization or visit to emergency departments. That happened for 5 patients among the cohort dosed to the 3 doses of the neutralizing monoclonal antibody compared to 9 in the placebo. And the percentage you can see is 1.6 versus 6.3. Obviously, the numbers here are quite low, and therefore any difference needs to be interpreted very carefully. The study was essentially not really powered for that. But it is the first publication that demonstrates, at least as a proof of concept, that there is a link between infusion of neutralizing antibody and biokinetics. The FDA recently announced an emergency use authorization for this monoclonal antibody. They are quite clearly articulating that there is a emergency use of this unapproved product for the treatment of mild to moderate COVID-19 in adults and pediatric patients with positive results of direct SARS-CoV-2 viral testing who are 12 years of age and older and weighing at least 40 kilos and who is at high risk of progressing to severe COVID-19 disease and hospitalization. So in other words, not every patient who is infected should even be considered to be using this agent if you want to follow this emergency use authorization possibility. But if you are considering, it should be those patients that is at high risk of further disease progression and hospitalization. As you saw, there were 3 doses studied in the study, and the choice, since there was not a dose response curve identified, to use the lowest dose, the 700 milligrams. The infusion time is around an hour, and again, emphasizing it's not authorized for patients who is hospitalized, people who is requiring supplementary oxygen, or people who are on chronic oxygen therapy and who requires increases in oxygen flow due to COVID-19. So what are the patients that is at high risk? This is obviously an area that is increasingly important if we are moving to early treatment also before hospitalization. So which are the patients we should be most concerned of? Again, obese people, BMI, high BMI, people with CKD and diabetes, people on immunosuppressive disease and immunosuppressive agents, and then older people by and large, and then we obviously is focusing on hypertension in older people as a risk factor on top of these, and also people with chronic obstructive pulmonary disease, and in younger people, in particular the teenager, those who are either having heart disease, sickle cell disease, and neurodevelopment disorders, so a quite selected group of younger people. Just to give you an update on another trial, this is the Regeneron cocktail of 2 antibodies. They recently reported that in a expanded press release. We're studying 2 doses of the cocktail, 2.4 grams and 8 grams. Again, patients who is a similar type as we talked about in the earlier trial as well. Here, the primary endpoint was change in viral load through day 7 rather than day 11 on the Lilly compound, and where they saw a slightly more robust decline in favor of those randomized to the Regeneron combination compared to placebo. There was also, again, a few patients who were in need for hospitalization and medical visits, more broadly defined, which was happening slightly more frequently in those who were randomized to placebo compared to either of the 2 doses of Regeneron. That combination have not been released by the FDA for emergency use authorization, as there are other data from that combination in people who is hospitalized that is creating some concern about safety for that agent. So the only one we have is the Lilly antibody. So just briefly, vaccines. I think we all want to see a vaccine, an effective and safe vaccine soon. And I'm sure we have all heard about the press releases that came out, both in terms of the Pfizer vaccine as well as the Moderna vaccine. Both of them are messenger RNA technology-based vaccines. Both of them are, in both studies, people are vaccinated at study entry, and then after either 3 to 4 weeks. Both included patients also who are older and at more at risk of severe disease in a sizable number of the population. You can see 41% in the Pfizer study were in the age group 56 to 85 years, and in the Moderna, over 7,000 patients out of the 30,000 enrolled in total were in risk because they're of older age. So these are two very large studies. Again, 43,000 patients in the Pfizer and 30,000 in the Moderna. Of course, to demonstrate efficacy, you need to have events in the cohort diagnosed. So you can look at whether the instance of those events are reduced in the vaccine group compared to placebo. Pfizer very recently announced that they have now 170 cases, and the interim data from Moderna is 95 cases. Importantly, the data that has been publicly announced so far are only looking at cases that happens within 7 days after the second dose or 14 days after the second dose. So really only the very short-term follow-up after the patients were enrolled, and at least in that time window, that fairly brief 1 month, 5 to 6 months, 5 to 6 week window, there was a pronounced reduction in incidence of SARS-CoV-2 in those who was vaccinated compared to those in the placebo. Importantly, one thing is whether it's preventing infection, but we obviously want to, in particular, see that the vaccine is also effective in preventing severe disease. 9 out of 10 cases from the Pfizer study were of the severe cases occurred in the placebo arm, and 11 out of 11 cases in the Moderna trial interim data also occurred in the placebo. So it looks like that there is a parallel reduction also in these more severe cases of infection. These are all press releases. We obviously need much more data in a proper scientific publication in order to really digest and fully understand the data, not only in terms of efficacy, certainly longer-term efficacy. Both studies were launched in late summer, and by definition, therefore, have relatively short follow-up time. But it's going to be important to look at whether this more than 90% efficacy is maintained also for longer-term follow-up. We want to study and understand the short-term safety profile. We have seen data from phase 1 and phase 2 trials for both of those vaccines, so we have a sense of the short-term, more frequent side effects. But obviously, we want to also understand long-term side effects. These are new technologies. There's not a licensed vaccine using this technology in other infectious diseases. So this is going to be critical to understand and study that, which I'm sure it's going to be happening. So there is a lot of vaccine trials underway. There is more than 87 who is simply preclinical, 38 in phase 1, 16 in phase 2, and 12 in phase 3. And obviously, we are waiting for the first vaccines to be approved. That may take a month, maybe 2, at least for an emergency use authorization in the United States. And we'll see how the other regulators around the world is handling this and how quickly they want to provide an equal sort of early approval for release. Again, durability is going to be critical to understand. We all have very short-term understanding of efficacy. safety, I cannot stress enough. Is this a vaccine we should use only for people who have not yet been infected once? There is a potential issue with reinfection that is emerging. So would the vaccine also be helpful in that situation? And then for these messenger RNA vaccines, there is storage and distribution challenges. Although it looks like that they are more stable also in refrigerators than what was at least initially reported. But that, of course, operationally, as you start to do vaccinations, we need to fully understand that. I think this was now time for questions. So Tiffany, I'll turn it back to you. Thanks.
Great. Thank you, Professor Lundgren, for that really comprehensive update and overview. So we've received a number of questions, and we'll do our best to get to as many as we can. So from Sarah, she wants to know how well the Moderna and Pfizer vaccine study arms matched for participants' exposure level to COVID-19. Can we be certain that the risk was equal in both arms?
Yeah, I mean, that's a very good question. And just coming back to the slide here that gives sort of the preliminary data here. I mean, we're talking about for the Pfizer 43,000 patients and only 170 of them have actually contracted the infection. So the incidence is obviously fairly low at the moment. And you know, the same numbers are true for the Moderna. It's obviously very difficult when you plan a vaccine trial to actually say, oh, we want to go into that population because that's where the pandemic is going to be raging, and therefore we will accumulate events fairly quickly. I think that has been a challenge. But obviously, the more follow-up there is, the more events is going to be accumulating. In terms of whether it sort of biased the results, both of these trials are double-blind and placebo-controlled, so none of the people who is participating in the studies will know whether they have been receiving the vaccine or placebo. At least by design, that's not going to be possible. So therefore, I guess it's a fairly safe bet. Again, we need to see the publication to really make that determination, but it's a fairly good bet that behavior is similar in the two arms of the study, and therefore the risk of getting infected and at least get exposed to the virus should be equal. Which obviously is the assumption we're making when we make determinations about vaccine efficacy.
Great, thank you. So Jessica asks, how many patients did not take the second dose and why? And I don't believe that was in the press releases, but could you maybe speak to the tolerability of the vaccines in the earlier trials? Yeah,
absolutely. No, that's a good question. I don't know the answer to how many were not dosed with the booster. I mean, the phase 1 and the phase 2 trials is coming out with a fairly consistent picture that in terms of safety, there's much less safety issues with the first injection compared to the second injection. The side effects that people are complaining of is fatigue, injection site reactions. Some have chills. So the sort of more typical vaccine responses and effects that you will expect to see for any type of vaccine. So that's I guess. So Tiffany, the other aspect of your question, I'm sorry.
I was just asking, and I think you answered it, just the general tolerability of the vaccines from previous studies.
Yeah, but one thing I just wanted to mention in addition to that whether, because there will be a debate, do we really need the second vaccine? And at least for the data that we have seen from the phase 1 and phase 2, the booster after either 21 days or 29 days creates a very high additional neutralizing antibody titer compared to only what has been seen up to the day of the booster. So it looks like from the data that you do need both vaccines to really have a good protective immune response.
Okay, thank you. So Cashel asks, what are the outcomes of the primary endpoint for these studies? Was it prevention of COVID disease or prevention of hospitalization in severe cases?
Again, we are only really gauging this from press releases, so I need to be, I cannot sort of fully explain this, but the intent was to take people who have not been infected before, who are well and presumably not infected when they get into the trial, and then look at and then vaccinate or enter them into the trial in this randomized way. As mentioned also when I went through this slide, there's at least an intent here to also include people who is at high risk should they be infected. And there is on the right column here, you can see that there were what was classified as severe cases, so infections in people who is at high risk of hospitalization. But I'm not sure that there will be enough power to really look at sort of the impact on more, ventilation, for example, or mortality related to the infection, because I don't think that there will be enough power to really decipher that. So I think we will need to run with understanding whether the vaccines is providing protective immunity. And one thing that I think which will be a critical point to focus on as these studies get further analyzed is whether it's possible to identify a minimal titer of protection. So is there a certain level that you need to achieve, and if you achieve that, then the risk of infection is essentially very, very small. That would be a very helpful information to get out of this, because we can then start to understand what is the kinetic of changes in the antibody levels and how quickly are people getting below that threshold, and because maybe we will need to do a third booster later on. So one of several questions. So this is really why we need to really make sure we fully understand for how long time is this protection actually happening. Thank you.
Okay, thank you. So we've had a couple of questions along the lines of the vaccine in persons that have previously had COVID. Amit asks, do you think that antibody levels should be checked before vaccinating? And Ryan asks, what about patients that have received some of the monoclonal antibodies, and do we know, if there's any concern about patients that have natural or have been treated with antibodies in terms of vaccination?
No, I mean that is I think one of the emerging critical questions to understand. Is there benefit of a vaccine in people who have naturally acquired immunity because they were infected, and if they received one type or another of passive immune therapy, being a neutralizing monoclonal antibody, as we discussed, there's obviously been convalescent plasma being used as well. Is that impacting in people who have recovered the COVID disease? Is that impacting if there is a benefit from using vaccines after the infection? Are still circulating antibodies due to passive immune therapy? Is that impacting on the vaccine response? I mean, these are all good, excellent questions that needs to be studied, but we don't have the answer to that essentially at the moment. But they are being discussed extensively and will be addressed.
Okay, thank you. And you kind of alluded to this, but Michael says that based on the efficacy of the two antibody treatments, what would be your thoughts about using them for high risk patients? You said you probably won't have access to it, but
what has been creating a little bit of a curve around these antibodies is that the Regeneron announced in a press release in the end of October that in their study of their antibody in hospitalized patients, they essentially had four sub-cohorts where they're randomizing the patients into, and cohort number 1 and 1A are people on supplementary oxygen in different degrees. Cohort number 2 are people on high flow or non-invasive ventilation, and then cohort 3 are people on ventilator. And what they announced here in the end of October was that their Data Safety and Monitoring Board have requested the pause of further enrollment into cohort number 2 and 3 out of a safety signal. So that at the moment, what is going on there? Is this a problem specific for this agent? Is it a more wide problem? Is under intense discussion and investigation at the moment. But I think that's one of the arguments for why you don't want to use these antibodies in hospitalized patients. The US NIH treatment guidelines as well announced that yesterday, that they don't see at the moment a role of a neutralized monoclonal antibody in the treatment of COVID-19. So I guess that was their position on that.
Right, that's important, and thank you, Robert, for reminding us to mention that. So we continue to get quite a few questions on the vaccine. We've gotten multiple questions on the durability, as you've already kind of alluded to, but are there any data from previous studies or from how long the measured immune responses seem to last that might be able to predict the durability of these vaccines?
I mean, the best data we have, of course, is the natural immunity and how quickly natural immunity, at least on the antibody side, the B lymphocyte side, how quickly are these antibodies declining? And they seem to decline at a fairly brisk rate over the first 3 to 4 months. Whether that's true for vaccines, again, whether they will have the same kinetic of decline, good question. They obviously seem to start from a higher level compared to patients who's recovered from the acute COVID-19. It seems like the vaccines are placing patients at a higher titer level, and therefore they essentially have longer to go if there is a decline over time. And then, of course, the whole other aspect of this is T-cell immunity, which is slightly more complicated to study. It seems like these messenger RNA vaccines are inducing T-cell immunity. It's unlikely that T-cell immunity, at least by itself, is going to be providing protective immunity like we are seeing on these very, very preliminary results. That must be really due to antibody response, at least most likely. But whether T-cell immunity actually are more robust and therefore maintained for longer periods of time, and therefore if you have generated that you can more quickly also reintroduce the production of neutralizing antibodies if you get exposed before you actually feel the infection is a possibility. But a good question. I think many would like to have the answer to that, but unfortunately, we'll have to have another discussion on that when there is that discussion is a little bit more informed.
Right, thank you. So Javier asks, what about the risk of immune enhancement disease post-vaccination? Is there any concern for that?
So antibody-dependent enhancement, obviously we know that for other infectious diseases, for example, dengue infection, it looks like those who have antibodies, they will have a worse disease course compared to those without. And the whole issue around vaccines in that for that disease. So we have examples of that. Whether this is a problem in COVID-19 at the moment, we really don't know. This is we don't have a strong suggestion that it may be a problem, but I think the most honest answer to that is at the moment that we need to study that. And if antibody-dependent enhancement will happen, it's unlikely to happen early on, because then in that situation the antibody titers are fairly high. But in the tail end, if antibodies are gradually declining and they come below that sort of minimal protective titer, but still there, at least theoretically, that is a possibility. I don't think we will expect it, but I cannot rule it out neither.
Thank you. So following up on that, Delia asks, though the pediatric trials are just starting, would there be any concern of the immune response triggering the multi-system inflammatory syndrome we've seen in children?
Yeah, I mean, I have to say that is a very good question, and that's probably also a very good argument for why we want to, at least, I know that the Pfizer is including children with 12 age or older, so we'll have some information from there. But I think there needs to be a very strong case to go out and start sort of mass vaccinate children until we have more data, because it looks like the immune response is different. It is different. Whether that's good or bad, we don't know. But we certainly, as being suggested and mentioned here, there has been some children after their acute infection that had a hyper-inflammatory response, and whether the vaccine is a problem, therefore, more so in children compared to adults. I think we just need to have more data before we can really make that determination. I mean, it comes back to the question around prioritization, because at least in many settings, there won't be enough vaccines to vaccinate sort of everybody who wants. So there probably needs to be some prioritization happening. And of course, the more vulnerable part of the population is probably the ones, as well as healthcare personnel, is probably going to be the two sort of sections of the population where priority should be given, at least initially. And then wait probably some time when we have more data on kids before we start doing that there.
Right. We need more data. That seems to be the...
Yeah, well, I mean, it's early days. I mean, it's...
So we have similar questions from Jim and Ralph about efficacy and safety in racial minorities and in older people for the vaccines. Ralph specifically asks, is there sufficient data to feel safe administering the vaccines to people of color and older individuals, in your opinion?
I have to say, I think both trials, and I also know many of the other phase 3 trials, is really trying to engage in the study population people of different ethnic background. You can see in the Pfizer study, 42% of the volunteers had diverse ethnic background, and similar, it was 37% in the Moderna. So we will have the possibility of looking in subgroups depending on ethnic background to understand both in terms of efficacy but also safety, whether there is any signs of differences depending on people's ethnic background, in addition to age and in addition to gender. So these subgroup analysis is going to be very important and very informative to get a better sense of that.
Okay, and we've had a couple of questions on this from Sarah and Lehana asking about what's the latest speculation on what percentage of the population will need to get a vaccine to reach herd immunity, or is herd immunity a long gone dream? What's the latest on that?
Well, I mean, there's two aspects to herd immunity. I mean, one is that in the population that is driving the epidemic, we want to have at least 70% of them immune before the chance of that population continue to drive epidemic spread of the infection is essentially nullified. That may not be needed in older people before, because they will receive the vaccine mostly to protect themselves, because they are typically not part of a transmission chain, but rather they are infected because of spillover from where the epidemic is mostly happening in the younger population. Yeah. Did I get that right? What was another aspect to this?
I know you got it. They were just asking what the percentage of the population should get vaccinated to potentially reach.
Yeah, so 70% is sort of the benchmark, but obviously anything also below 70 is not going to be eliminating the risk of an epidemic in that population, but it will certainly reduce it. I mean, obviously that's why we, in our annual seasonal influenza outbreaks, they're not larger than what they are because of immunity in the population, not sufficient to completely prevent the influenza outbreaks from happening, but nevertheless it's there, and therefore they don't get not more people get involved with them. So I think the more we can get people vaccinated, the better it is, and the more "quote unquote" normality will be reintroduced into the population.
Okay, and I think we have time for one more question, and we've had a couple of questions on this topic. G asks, would there be any concerns giving the vaccine to patients who are on immunosuppressive therapy or patients that have an immunosuppressive disease?
I think the concern is mostly whether the vaccine is actually going to be working, because there is not sufficient number of B cells to be activated. So in terms of safety, probably not, because I mean this is just messenger RNA. So we are not talking about a live attenuated vaccine. So from a safety point of view, I don't at least immediately can see a safety issue. But one, I think I would be concerned about whether the immune response is wanted. It probably is.
Well, thank you very much, Professor Lundgren, and thanks to our listeners. As a reminder, to view the COVID-19 Comprehensive Resource Center on the Clinical Care Options website, click on the link in the show notes. Please be sure to check back regularly for more episodes on important infectious disease topics. Thanks!